bed. Everywhere you look, there's a heart, a hand to hold on to. Everywhere you look, there's a love.
And on today's episode, we're going to be talking about mitosis. We start the cycle with a diploid cell, and it begins growing in the G1 phase. DNA replication occurs in the synthesis or S phase of the cell cycle. In this process, the double-stranded DNA is unwound, and each individual strand is used as a template for the production of the complementary strand. The end result of the production is two identical copies of the genetic material that we call a homologous pair. In this homologous pair of chromosomes, we have alleles. An allele is a pair of genes that appear in a particular location on a chromosome that control the same characteristic. After the homologous pair connect, this chromosome includes a telomere, which is a region of repetitive nucleotide sequences at the end of the chromosome. Telomeres function to protect the ends of chromosomes from sticking to each other. They also protect the genetic information from being lost during the replication process. Next, we have the centromere, a specialized structure that appears during cell division. This is the central region where the two chromatins are held together to form an X shape. This is also the region of the chromosome where the spindle fiber is attached. Around the centromere, you see the kinetochore. This is where the microtubules of the spindle fibers attach. The two halves of the chromosomes are the sister chromatids. After the S phase, or the synthesis phase, the cell continues to grow in the G2 phase until it is ready to completely undergo mitosis.
In interphase, the cell is undergoing normal metabolic activity and preparing for mitosis. The two centrioles are present in the cell, close to one another. The nuclear membrane is also present. Chromosomes are in the relaxed form called chromatin, making them difficult to visualize under a microscope. Also, the spindle fibers are formed from microtubules, which we call centrioles. In prophase, the chromatin in the nucleus begin to condense and become visible under the microscope as they appear as individual chromosomes. Each chromosome possesses two sister chromatids, which are attached together by a centromere. Centrioles begin migrating to opposite ends of the cell and extend their microtubule fibers. The mitotic spindle is then formed. In metaphase, chromosomes line up on the metaphase plate, which is the equator or middle of the cell. This happens with the help of the mitotic spindle, originating from the microtubules of the centrioles, which are attached to the kinetochores of each chromosome. This organization on the metaphase plate ensures that each new daughter cell will receive one copy of each chromosome. In anaphase, the paired chromosomes are separated at the kinetochore into sister chromatids, which are pulled by the spindle fibers towards opposite poles of the cell. This mechanism results from a combination of kinetochore movement along the spindle microtubules and through the physical interaction between polar microtubules. In telophase, the chromatids arrive at opposite poles. The nuclear membrane then reforms at each pole around the newly separated chromatids. At the same time, spindle fibers are dispersed in the cell. Chromatids relax again and take on the form of chromatin. Then, in cytokinesis, newly formed daughter cells are eventually pulled apart as a result of the contraction and pinching of actin. Thus, two new identical daughter cells are created, concluding the process of mitotic cell division.